As Dr. I've said, I am a rising fourth year at Scripps Research. I am in the Petroshek lab. And today I'll be giving you a talk titled Using the Power of Chemistry to Target Age-Associated Diseases. And I'm going to spend most of my time actually talking about what it's like to be an Indigenous man in science, how that affects my science, and how it affects how I do my science, because I think that's the most fruitful thing that we can get out of all of this interaction that we're having right now. So I'm actually going to start with a land acknowledgement. I want to start by introducing myself in relation to the land that I come from and where I am now. This is out of respect for my ancestors, as well as the Kumeyaay people whose land I'm currently on here in San Diego. I think it's important to acknowledge that as an Indigenous person, I have a particular relationship and situated knowledge to the land and its peoples, and you'll hear a lot about that in my research. This knowledge shaped my journey into becoming an academic and informs how I do my science. So for my own creation story, by the way, a creation story is just a story that describes how things came to be. Starts here in this little blue dot, which is the White Mountain Apache Reservation. This is probably a map you've never seen before, so I actually took the liberty of poorly drawing Arizona on it. And so obviously California, Arizona, New Mexico. So I'm from Eastern Arizona. And in Eastern Arizona, there is essentially the site of creation. This is for the Apache people, Mount Baldy. And this is the land where I'm from. Since then, I have moved, obviously, now to San Diego, which is in Kumeyaay territory. And this is actually a picture of two Kumeyaay women who are at the border that I drew in here, protecting sacred sites from destruction by the Army Corps of Engineers to build space for a border wall. But I'm going to reference this photo pretty often through this talk, and it's really to highlight and show how chemistry intersects with science and society, and how that also shaped my journey and how I do my science. And if there's one thing that I want you to take away from this talk, it's that I'm going to be discussing my journey linearly, right? But it's actually way more messy and complicated than that. I think that's highlighted really well with these two maps, right? So on the top here, you see very structured, organized borders between Arizona and New Mexico. Apparently, Mexico doesn't exist, as opposed to this land of relations here, where you show a very kind of messy, overlapping, always changing web of relations, as I kind of refer to it here. And we'll be talking about that a lot too. So I just wanted to bring that out there, and that's how it shaped my journey. It's going to be described linearly, but it's more of a web. Educational opportunities often are divided by race and class. These are two pictures of my my grandparents here, my grandpa on the left, my grandma on the right. And they raised me on a small reservation on the Fort Apache Indian Reservation, which is shown here. But educational opportunities aren't equal amongst everywhere, especially across space. So this red dot here is an off-reservation high school, which is about 50 miles away from where I lived. And the thing through different colonial mechanisms that happens is that the off-reservation high school had far more resources in terms of AP classes, funding, pre-college classes, all of that kind of stuff. And so for me to be successful in college, it was ideal that I would be able to attend that. But through certain legislation mechanisms, you have to live within the school district. And being 50 miles away, I wasn't within the school district. But I found a way to circumvent that. And that was by being adopted into my best friend's family. So that's them right here on the left. Obviously, I stick out a little bit, but that's my best friend here, Kenny, on the right. And I was able to build this network of relations of kinship, as I refer to it up here, in order to begin building capacity to end up at Scripps like I am now. That started very early on by building these relations. Anyway, back to my grandparents. So this slide is titled Movie Magic, Chemistry and Cinema. And the reason for that is because my grandpa, when I was growing up, always loved to watch Western movies, while my grandma always loved to watch corny 90s action films. So like think of like Arnold Schwarzenegger films, or like Jean-Claude Van Damme, things like that. Anyway, one of the common tropes here always, and especially was like huge for me as a kid, was explosions, right? This often are catalyzed by this chemical called nitroglycerin, trinitroglycerin, TNT. I drew the molecular structure here, and I took this photo, actually, of dynamite in the background. And this is at the Kumeyaay land border, where these two land defenders are protecting their sacred sites. So TNT can evoke, or chemistry generally, can evoke this imagery of explosions and destruction. But what's really interesting and powerful is that it can also evoke medicinal properties or healing or restoration. TNT is a potent vasodilator and was used as a medicine for many, many years in conjunction with it being used as an explosive. And so I think that highlights this really powerful implication in society that chemistry has between destruction versus restoration. And sorry, one last thing. In this whole sphere is like what got me started in chemistry was just being a young kid who liked explosions, but it turned out to be so much more than that. This is another map where I moved from Eastern Arizona to Southern Colorado. And this is me expanding that web of relations, kind of, growing out, so to speak. And I went to undergrad for my first year at Fort Lewis College, which is depicted up here. This is known as a land grant university. So in exchange for typically stolen land, Indigenous students get to attend this college tuition free. There's obviously all kinds of other fees and things that come with it, but this was an educational opportunity that was afforded to me so that someone from a lower socioeconomic class was able to access higher academia. There, I was a chemistry major, and I did specifically organic chemistry research, where I researched near-infrared dyes for biomedical imaging. This is a structure of one of the dyes, and it's based on indigo, where you make cheap, lowly expensive, low toxicity molecules that you can swap out, say, these fluorines amongst this indigo structure and tune the dye to have different kinds of physical properties. What we were gunning for was to develop a bioimaging agent so that you would be able to visualize the vascular structure of mice brains. I was only a freshman in college. I didn't really get that far with this project, but it started me on the path of synthetic organic chemistry and giving me the basic tools to go about doing it. From there, though, I was really lucky and fortunate to receive the Gates Millennium Scholarship, which is a full ride scholarship to anywhere in the US. And what this did is it gave me some of that socioeconomic capital to attend another better school that had more resources. I loved Fort Lewis College, but visiting Los Angeles, which I'll talk about in a second, was really key. I went to Loyola Marymount University. That is shown here. It's actually one of the top-ranked film schools, which is kind of interesting to tie back to my grandparents' love for film and how that instilled that initial chemical curiosity. It seemed like that always has been following me through my life. Also, my brother went there. That little white boy that I showed you before, he's one of my kin, right? He's my brother. He helped me get into the new school district with the better resources. And he also helped bring me to LMU very much in the same way. And when I was visiting him one time, I saw that LMU was building a really beautiful new life science building, super gorgeous. And while I was there, also, like, Los Angeles is, one of the social hubs of like culture and film. And so it was really great for me to be able to be introduced to that as a small mountain boy. And all of those things kind of drew me towards it. So I did do my 3 more years of chemistry education there. What I was primarily focused on researching was this here, which is a G quadruplex structure. Basically, you have DNA that's going along, beads on a string, and it can fold and stack upon each other. And certain bases, particularly guanine, can stack on each other on this G quadruplex structure. And this is really interesting because it can form a sort of knot. And when you stabilize those knots with molecules like ligands, you can inhibit telomerase. I don't have time to really talk about it, but this is a therapeutic strategy for anti-cancer drugs. And so we were trying to recreate that in a molecular fashion. But broad strokes, general thing that I brought with me through my research now and Scripps is that I was able to use chemistry to answer biological questions. And then I took 2 years off between undergrad and graduate school. This time I'm going to summarize in just this one slide, but please ask me about it if you're really interested in taking some time off between undergrad and grad school. But there's just so much to unpack that I gotta keep it short. So I got a lot of research training though, and when I was there, I was able to study for the GRE, which was really huge because I took 2 years of studying, right, and was able to take the tests multiple times because I did not score very well on those tests, and I'm not afraid to admit it. And I think one other thing that was really big for me is not being poor. Going from like generations, intergenerational poverty to being able to someone that had a little bit of social capital and socioeconomic capital, like that was huge for me. And then I was able to have different kinds of hobbies. For example, I was able to go rock climbing. And in this way, chemistry allowed me to reconnect to the land, right? This is a very visceral way of connecting to the land after being gone off the reservation for so many years, because I'm just physical hands on the rock out in the mountains and stuff. That was really great and was given to me by pursuing this chemistry. In short, like Professor Ive said, I worked at Bachem where I did solid phase peptide synthesis. And then I also worked at Hologic where I did analytical method development, basically making molecules and then analyzing them through different statistical standards and methods to ensure that they're pure and they're doing what we mean for them to do. Again, please ask me about this if you would like. This was a huge part of my life, but I don't have time for it. From there, I started at Scripps. This is going to be my first year where I was really sharpening my old skills. Like I said, I was working peptides in the industry after undergrad. And so I jumped right into a lab at Scripps and started doing peptide synthesis and was able to get 2 peptide papers out of it and just generally further my training in organic synthesis. But I think what's key is I wanted something more, right? Like rock climbing allowed me to reconnect to the land in a visceral way, like I mentioned. How can I reconnect to my community in a different way through my chemistry, through my research? I think that brings us back to TNT. For TNT, it can be used for destruction and restoration. Like I said, it was used to demolish sacred kumai sites. Actually, I took this picture like I was sitting in my car and it was just right 2 feet outside of my car. This is through my window. But this is the kind of premise that sounds straight out of some bad action movie that my grandma would love so much. And we were able to flip that on its head, right? Like it's destruction, but also TNT can be restorative through medicine. And so how can you take chemistry and reimagine it as an instrument for my community? That statement is very bold and out there here. And also, it also has different kinds of implications with indigenous sovereignty and indigenous-led research, which you can ask me about also, but I just don't have time to really unpack all of this stuff because I want to talk about the science as well. But the past 12 minutes have been meant to build a framework of who I am and how that's going to affect my research, which I'm going to talk to you about now. I study age-associated diseases. On the left here is a mortality disparities rate chart provided by the National Institute of Health. This is a chart that documents different mortality rates in indigenous populations. And I tried to distill this all down into one single sentence to be able to inform my research. Because of settler colonialism and institutionalized racism, indigenous people have a life expectancy of 5.5 years less overall than US populations. The main drivers of these are heart disease, cancer, and diabetes. Those are the top 3. But all of these are known as age-associated diseases. What an age-associated disease is when you get older, as depicted here on the x-axis with age, the probability of you dying from the disease increases, as depicted here on the y-axis. That's true for heart disease, it's true for cancer, it's true for diabetes, and it's also true for Alzheimer's disease. There are tons and tons of treatments that look at disease-specific ways of intervening. So think about cancer. There's all kinds of chemotherapies and so on and so forth, right? Those are all disease-specific, but again, my lab tries to flip that narrative and ask what are the common mechanisms that underlie all of these. And I think quite obviously, like it's right there, it's age. conceptually, we can think of age as increasing disorder to death. This is a really like intuitive graphic my own advisor made, where you have a very nice structured ordered grid, and then it starts to get a little dysmorphed and finally starts to break. There's some damage in it, and then ultimately all of that accumulates and leads to death. My specific research is focused on Alzheimer's disease and similar diseases like it. These are known as neurodegenerative diseases. So neurodegenerative diseases are a subset of age-associated diseases. And so now I'm going to talk more specifically about this neurodegenerative disease. What underlies neurodegeneration? What are the mechanisms that drive it? This is a really nice picture from Dr. Giovanna Malucci. On the y-axis is the incidence rate, and on the x-axis is age, right? And what happens is the older you get, your synapse number starts to decrease. A synapse is a structure that permits a neuron to pass a signal from one to the other. So essentially a way of communicating. And these connections, this web, again, allows us to make memories and maintain a healthy brain. So that decreases with age. Misfolded protein accumulation increases with age. This I kind of loosey-goosey refer to as aggregation, like the accumulation of these proteins into aggregates. What I mean by that is very clearly depicted here. So in a normal neuron, you have a nice, healthy, spindly dendrites coming from a cell body. However, in a diseased neuron, you see that there are the accumulation of these plaques, these senile plaques and neurofibrillary tangles. This is specifically Alzheimer's disease, but it's true across all, it's true across all neurodegenerative diseases. There's increasing in misfolded proteins accumulating in an aged brain or a diseased brain. And all of that accumulates into this graphic, which I think is like super, super clear, right? On the healthy brain, you see everything is nice and big, whatever. And on the severe AD, there is literally neurodegeneration, and that is what neurodegeneration is. Aging can drive neurodegeneration. How do we study these mechanisms? I use a small non-parasitic nematode called C. elegans. They do not look like this, but this is a really cute cartoon of one by Ellie Elegans from Twitter, and I'm going to use a couple of her pieces now. Each single worm has an average of about 300 progeny, so they have 300 more babies. They are hermaphroditic, they self-fertilize, and so this maintains a very even population of genetically controlled offspring. They're also very small. They're only about 1 centimeter long, which is about the size of a comma on like a normal book. And that makes them really easy to maintain and really easy to house. All I do is I keep them in little petri dishes about that big. And this is a really well-established model for aging research, health research in general, that's helped win 3 Nobel Prizes already. And so like this is a very good model to study aging. On the left is a picture of a normal healthy adult C. elegans, and when they age, they do not grow a beard or have a cane, but they do elicit certain phenotypes that I'll show you in a minute. And that's essentially what I do is I grow up these worms and I see how they age. The general guiding principle of this is to ask, if we delay aging, can we delay age-associated disease, such as neurodegenerative diseases or heart disease or cancer? And the answer is yes. What I'm depicting here is a survival graph. So on the y-axis is the fraction alive of C. elegans over a given time, which is depicted on the x-axis. So at 1, 100% of the worms are alive, and at 0, 100% of the worms are dead. And in red, you see that this is an untreated worm population, and half of the population lives about 10 or 11 days. When you elicit a particular treatment, which I'm going to show you on the next slide, these worms live 73% more. They live way longer, which is what I'm trying to get at, right? Like, we're delaying aging. It turns out that by delaying aging, making things live longer, you can delay the onset of age-associated disease. This is true for worms. This is true for flies, mice, and monkeys, and some other kind of small rodents. But this is conserved evolutionarily. So I think this is really exciting place to do research right now. That particular treatment that I showed you on the last slide was giving minocycline to the worms. This is the structure of minocycline right here on the left. It's a second generation antibiotic. It's FDA approved for acne vulgaris, and it's beneficial in many models of Alzheimer's disease. So again, like it's delaying the onset of age-associated diseases, so maybe it's delaying aging itself as well. What it does is exactly that. My lab studies this molecule. If we conceptually think about aging as increasing disorder to death, it slows down the disorder and slows down the damage and ultimately leads to a delayed death, delayed onset of neurodegenerative diseases. How I study this molecule and molecules like it, I'm going to show next in this experimental setup. C. elegans hatch into a little egg, and they go through various larval development phases, which is depicted by the L. But basically, about after 3 days, they become fully reproductive adults. And these adults are reproductive for about 5 days, and then they go on to live, however long, usually a maximum of about 30 days. And what I do is I give treatments to these animals when they are adults, so I don't interfere with the development of the larvae. And I see how long they live. What's really interesting, though, is asking, what about in aged adults, right? Because you're not going to give an 18-year-old a drug treatment and say, hey, take this for the rest of your life. You're going to have to give it to aged individuals. And so we tried to recapture that in C. elegans by giving it to them in day 8, which is like quite old for a C. elegans. They stopped having kids several days ago. And it turns out that out of all of the molecules that were screened, only 1 or 2 actually work in aged animals in the yellow. That's shown here, where black is untreated worms and red is worms treated on day 1 when they're young adults. They live about 48%. However, when you give them this drug on day 8 when they're old, they live a little less, but it's still, they're able to live, which is unique and special to this one specific drug. And then I want to take that a step further. Since I am trained as a chemist, how can I design new molecules that also extend lifespan, but better? And there's also more reasons for that. Minocycline is an antibiotic, and you can't take this chronically, right? Like, you're going to mess up the good bugs in your body, and it's going to lead to antibiotic resistance. There's just a whole lot of reasons why we don't want this to be an antibiotic. And also, we want to know if we can improve this molecule or maybe even combine it with other drugs to rationally design a greater lifespan extension. And that's exactly what I've been able to do and exactly what I continue to do now in my research. I can't show you the actual structures of these molecules because for intellectual property reasons, but suffice it to say that in black, you have your water treatment. In red, you have minocycline treatment. And then I found this one molecule that extends lifespan about twice as well as minocycline, about 60%. And then I found that a combination of these 2 molecules that we understand very well, when you combine them, it extends lifespan up to 79%, which was huge for C. elegans. This is like a big deal, and no one chemically has been able to create this big effect change in C. elegans. So the short answer is yes. And now I'd like to show you a little video. This is mostly just an interesting thing for me. It's kind of spooky. So at day 35, these untreated worms on the left here have been dead for about 2 weeks. And their treated friends on the right are completely fine. This is what an old worm looks like. So he's kind of in the in-between, but the other 3 are completely chill. Like, it's kind of weird to see. Yeah, so that's what the worms look like. That's them moving and behaving normally here. This is an old worm that's starting to move less and not be as happy. And then these are very dead worms. They've been dead for 2 weeks. And it's just a weird experience to have, but it's also like kind of cool. So that's like the age-associated disease side of things. I also work on developing chemical tools to study protein aggregation. So we can do some straight up hardcore biology right now. Protein translation. This proteins are the biomolecules that do all the heavy lifting in your body, right? They create things, they help you move, so on and so forth. They're translated, they're created by a ribosome attaching to an mRNA and slowly ratcheting along it and spitting out the top of it a unfolded protein, which is depicted here with these little dots. That's a nascent polypeptide. Once you get a full protein, which I've depicted here as like a spaghetti noodle, it can be folded and unfolded into a proper biological protein that does whatever function it needs to do. However, sometimes, especially when you get older, it can misfold into a misfolded protein, which can't do the biological functions it's meant to do. Also, it can be, these unfolded proteins can self-associate with each other and form beta sheets and other kinds of aggregates. That's what I'm referring to as folding and trafficking of proteins. And then finally, all of these proteins can be cleared through different kinds of mechanisms. One of them is the proteasome, which is essentially like a garbage disposal. It has these proteins that kind of spin along and munch up a protein and spit out the little amino acids at the end. And you also have autophagy mechanisms that envelop proteins and basically dissolve them. Very fascinating stuff. But protein translation turns out to be the biggest load on this system. So I think the obvious thing to do would be to ask, why not decrease this load, right? And that's basically what I do. I look for molecules that target translation to restore proteostasis. Returning to the networks of interactions, this whole interaction between translation, folding and trafficking, and clearance is referred to as proteostasis. And that's what I'm trying to target. I'm trying to find molecules that restore proteostasis in aged animals. This is what that looks like experimentally. I'm going to show you the live aggregation dynamics of C. elegans. So what I did is, well, I didn't do this. This was done by Richard Morimoto like 20 years ago, but he fused a fluorescent protein that we can see with a aggregation-prone protein, one of those proteins that like to self-associate and form aggregates. And that particular protein is called Q35YFP. It's more or less a model for Huntington's disease, which is another neurodegenerative disease. I take some C. elegans, grow them up on their little petri dishes, and then I subject them to a heat shock. I stress them out for 2 hours, and then mount them on a microscope slide and image them over 15 hours. And what you see, it's kind of hard to see in this, but there's a diffuse signal around the black of the worm. And then those signals kind of localize into little dots. Puncta is typically what we refer to them as. And that puncta are they behave like aggregates. For the best that we can tell, those are aggregates. Aggregation is forming. And that's what I'm depicting here. So after a 2-hour heat shock, gradually these puncta form, and you see strong aggregation in the heads of these models for neurodegeneration in these specific C. elegans. So then I had a system to study and hunt for these molecules, and so that's what I did. The particular molecules themselves aren't important. Suffice it to say that they're ones that inhibit translation. Like I said, that's the biggest load on the system. That's what I wanted to target. And I found one that works quite well. It's called anisomycin. You can clearly see here that there's less puncta in these worms. Anisomycin structures here for the organic chemists in the audience. And it turns out that they actually survive really well too. So I take C. elegans and I subject them to a non-permissive heat. They like to live at 20 degrees Celsius, but I hit them at 37 degrees Celsius and basically cook them and count how many until they die, how many are wiggling until they die. And what you see is that in black, the percentage alive decreases pretty strongly in untreated worms. However, in pink and in green, the worms are more resistant to this heat stress. And that's true for 2 very different structurally unrelated translation inhibitors, which means that we're onto something quite exciting there. And one last thing I want to show is another video that shows the aggregation dynamics happen in real time. I think it feels a little more real if you can see it happen with your eyes as opposed to a graph. On the left are untreated worms, and on the right are these anisomycin-treated worms, these translation inhibited worms. And you can see, sorry, I have the screen over here. You can see that these guys right here are starting to aggregate quite strongly, 14, 15 hours, as opposed to here. This particular worm has a little bit of aggregate, but nothing compared to like the ones on the right. And then these two up here aren't even aggregated at all. So that's generally what I do in terms of fluorescent microscopy to screen for new molecules. And last, I just want to talk a little bit about why and how this happens, right? Like what is underlying the reason that protein translation can be beneficial. And I think it has a lot to do with balance. So in young animals, you have a balance between folding requirements and folding capacity. That proteostasis network of interactions is in check. It's in balance, right? However, when you get old, it becomes out of balance. Your requirements are way greater than your capacity to fold proteins. And what these translation inhibitors do by decreasing load is restore that balance. It rebalances the folding requirements with the protein folding capacity, and so that the worms are able to be nice and healthy, like I pretend to show here by climbing a rock. And with that, I just want to summarize by saying that mentors are key. Here's some of the people in my life that have been instrumental in me becoming a graduate student at Scripps Research. Programs provide a lot of opportunity. Feel free to ask me about any of these programs, like the Gates Millennium Scholarship, McNair's, like Professor Ife mentioned, and NSPIRE, obviously. These are really provide resources that allowed me to be where I am now. And finally, I just want to leave off by saying, reimagine what chemistry can be, or what science can be, using chemistry to answer biological questions. This is in my own iteration of it. Delaying the onset of aging may delay the onset of age-associated diseases. Again, if aging is increasing disorder and damage until death, we're looking for molecules that inhibit that. And with that, I just want to acknowledge my lab, my committee members, the NSPIRE committee for picking me and choosing me to be a fellow, our funding agencies. This is my Twitter handle in case you ever want to hear discourse on indigeneity in chemistry. ChemCat is the handle. And finally, I want to acknowledge the Kumeyaay people who are still out there fighting the good fight and defending their sacred sites. Theoretically, it's been very great to be out there with them and standing in solidarity. And thank you all for listening. And with that, I'll take any questions.